Okay, everyone, it is 3:30. So for those of you that don't know me, I'm Professor Ron Lasky, and I have the privilege of introducing my daughter, Jessica, who is a associate professor at the medical school at Harvard. She is a graduate of Cornell University in biostatistics. She has a statistics degree, master's from Purdue, and a doctorate degree in the Harvard Medical School in
Epidemiology.
Epidemiology. I always have to think about that. So she's going to talk to us a little bit today, and thank you for attendance.
Hi, everyone. Can you guys hear me okay? It's super exciting to be here, and I'm excited to be talking about one of my favorite topics here, which is aging and thinking about how we age and the underlying mechanisms of aging. And let's see, how do I get this to go forward? Oh, there we go. And so just to open up, when we think about aging, right, I mean, I'm assuming people here have been sort of in the know and seeing that longevity is a key buzzword these days, right? We're hearing about it a lot in various frameworks. And really the premise is we have often thought of aging just in terms of the chronological numbers of our years. But as we are looking more into the biology of it, we are really understanding that aging is really a biological process, and we can quantify it and understand it in several ways. And one of the ways we can think of aging most broadly is really in a perspective where aging is really looking at the totality of our health, right? From how we can move to how we think. And the way that we age is really dependent on so many different things. And so with that framework, it gives us this different perspective of chronology with biology, where we can understand that the way and the process by which we age may be impacted by good things like exercise, the right diet, sleeping enough, and the bad things, right? Whether it be things that we do like smoking or being obese, drinking, but also exposures we may not even know we're exposed to. And all of these things build into basically the chronology of the health over our life. And in thinking about this as someone who is an epidemiologist and interested in health fundamentally, I think one of the things that is really important to talk about is what the current approach that the medical system basically has when it comes to health. And that is, the current approach is a very reactive approach, and it's really treatment focused. So people go along, right, and you get sick, you may get sicker, you may stay at home, oh, I don't want to go to the doctors. By the time you go to the doctors, very often, you are really not in a good physical place. And whether this is with a cold, but I'm thinking more so along the development of chronic diseases, and that the doctor then looks at you and says, you have diabetes, we're going to give you these different medications, but it is a treatment-focused approach. And so if we look at this in the perspective of just what's happening, right, the thing I want to point out is if we are looking at our total lifespan, with the treatment-focused approach, the time that we are sort of at optimal health, let's say we're 100% health span, is relatively short. And then we just keep on going down, and we don't actually get help until we're in a relatively bad place when it comes to health span. And in fact, now with all of the development of technology, what have we done? We've actually made it so, we've prolonged life. But the way that we've most often prolonged it is we've prolonged a period of life that is relatively low quality. It is not a time where you're 85 and you're running marathons most of the time. It's a time where, you may be older and have a lot of different health issues, and it is just not as quality and enjoyable as if you had a 100% health span. So, in thinking about that, I want to talk about the new paradigm of how we think about health along this process. And rather than being reactive, we want to be proactive. And rather than being treatment focused, we want to be preventative focused. And the reason for that is fundamentally, we want to stay at the highest level of health span as much as possible. So meet AI Jessica. So this is the AI version of me. And what I'm telling you is I'm not interested in a lifespan if this is how I'm going to end up for the last 20 years. What I'm really interested in is a lifespan where I am pretty much able to function the same, and my quality of life, the things I'm able to enjoy, are very high. So this gets to this concept of not just expanding longevity, but expanding health span, this optimized function, optimized health for the pretty much all of your life, right? And so in thinking about how we do it, it fundamentally shifts the paradigm in many different ways. And one of the big ways that it shifts the paradigm is to start early. And when I think of starting early, right, how early is early? Well, I would say early as you can get, right? And some of this, of course, we know cumulative habits, cumulative behaviors. But especially if you know you're disease prone in some way or another because of your background, right? Being aware of that and preventing it as early as possible, way prior to any symptoms, is really critical. And so the other thing is, if you start realizing you're having some type of decline in your health, intervening and starting the intervention when it's barely noticeable is going to really extend the health span period rather than waiting until things are sort of dire. And this is a framework, I think, that really, if we all live in, is going to give us the highest quality of life. But when we're thinking about that framework, and I'm thinking about that framework for one person versus another person versus another, a fundamental part of that is that we are not all the same, right? Not only are we different ages, different sexes, but we have underlying genetic predispositions. There may be certain things about you or certain things you've endured that may be different than someone else. And so our approach to how to do this really needs to be tailored to who we are if we want to optimize it. And one perfect example is nature versus nurture, right? Some of us have natural predispositions, genetic predispositions toward one thing or another, and that is going to have an important interplay on how we end up living. And here's an example I just want to show here. So this is, I think, around 2000, so about 25 years ago, me and my family. I'm a bright spring chicken, so is Dr. Ron. And this is my great-grandfather, who lived in 3 centuries. He lived to be almost 108, and remarkably was amazingly physically functioning the entire time. I think he stopped driving at like 98. He probably should have stopped driving earlier than that. But what was also remarkable about my grandfather is he had horrible lifestyle habits. I mean, he chewed tobacco his whole life. And he was a coal miner and had black lung, which most people die of at like age 35 to 40. So I'm really hoping that I have Grandpa Cherkowski's genes for longevity because those are really, really strong. But my point is, right, that is my background, which is totally different than other people's. And so incorporating that is really important. So this gets into the concept of how we think about aging and how we think about intervening, and a concept of precision aging or personalized aging, where you take a person, right, and you're really evaluating the totality of who they are on multiple different spectrums to understand what is biologically best for them. And now we already see this coming into play, right? Everyone has smartphones. I bet a lot of people have their watch on to check their steps. We have our sleep devices, et cetera. But on top of all of those things, I think really the area where there's a lot that can be done is in what I have over here, which are the different types of molecular data that we can look at that are really telling us critical things about our health that we may not be able to see from just smart devices, et cetera. And so we're going to talk a little bit about these and how this impacts aging overall. And so this is, when I think of aging, sort of a visual something like this of like, right, thinking about the different ways we can intervene, and there may be different areas or different things that we need to focus on for different people, or a combo of things for different people in different areas. So as an epidemiologist, right, if that's where we know we're going, and it's a super exciting place, one of the questions you can ask is, How do we get there? And I'll ask that now. How do you think we get there? What would you do? What do you need? Dr. Ron said it. Data, right? We really need a lot of data and a lot of information from a lot of different sources. And this is one of the things I think we can underappreciate. I remember sitting at Harvard School of Public Health when the Human Genome Project just came out, and Eric Lander was speaking about the first full human genome that was sequenced. And this, I saw in an article, I think this is like the 30th anniversary of the Human Genome Project, and a quote here by Richard Gibbs that says that the success of the Human Genome Project should not be measured by this. The success of the Human Genome Project should be measured by how the project transformed the rules of research, the way of practicing biological discovery, and the ubiquitous digitalization of biological science. And I truly believe that at the end of the day, this was really a transformative moment in how we looked at biology. And it is the concept of big data, right? And we see big data everywhere now. And the question is, when we're looking at something like aging, what type of data do we want, right? And the answer is, we want a lot of data. We want a lot of data on a lot of people. We want a lot of large cohorts. We want clinical trials, et cetera. But within that, we want all sorts of information, right? We want your step counter. We want to know if you've smoked. We want to know all sorts of things about your medical history, exposures, clinical labs, electronic health records, food. But then over here, what I have is a lot of the different types of molecular data that we are interested in looking at. And that's what I'm going to be talking about for a big focus of the rest of this session, is the types of molecular data that we're interested in and what they can tell us about aging. And where do we get that? Right? One of the places that my group has really focused a lot of our research to get that level of data is in biobanks. And for us, it's the Mass General Brigham Biobank that they have through Brigham and Women's and Mass General, which has about 150,000 people who've donated samples while they're healthy. But the great thing about this is, when these samples are donated, we can look at them in combination with other data, but we can measure all sorts of this underlying molecular data, right? And this molecular data is really going to be key into understanding how we can develop precision medicine approaches for aging. And again, that in combination then with electronic medical records, clinical diagnoses, clinical labs. Now, I'm going to spend this time talking about 3 different types of molecular data. And really, these first 2 are just to give you an idea of the types of things that are being done and what we've been able to measure with them and how that relates to aging. And then I'm going to focus on this last area because I think it's an area where there's a lot more that we have to understand and to learn. Now, when I have this top picture here, and I have DNA next to it, who knows what that is? DNA methylation. I see shakes of the heads and DNA methylation. Yeah, so DNA methylation. And what exactly is DNA methylation? What types of questions can we ask with this? One of the first questions that was asked, I want to say it was like in around 2012. I remember being at the American Society of Human Genetics when this paper was presented, was, okay, we have this concept of biological age, but are there actual ways we can measure it? And Steve Horvath, who did his postdoc in the same lab I'm in at the time, had uncovered some really important discoveries here. But I first want to just talk about generally what is the epigenome, and DNA methylation being one of the types of epigenetic modifications. So epigenetic modifications are, you have your genes are staying your genes. They're not changing. But the epigenome is telling your body how to turn on and off genes. So it's telling your body what genes to read where. And so if you have lung tissue, genes that are turned on are different than if you have brain tissue. And the regulation of that is really critical. And it's a critical component to aging. But along the way, different exposures, even inheriting your epigenetic imprinting from your mother. All of these things affect what is turned on and off. And as you age, the epigenome can change. So this over here, let's say, is what we're going to call a hypermethylated part of the genome. Now, not everything that changes from hypermethylated to hypomethylated is always bad, but predominantly as you age, hypermethylation changes to hypomethylation. So you have AI Jessica here looking really, really good, right? And so what happens as the hypermethylation changes over time, right? Different spots slowly start turning off. And as things are slowly turning off, AI Jessica changes a little bit, but then it keeps on going and more things change. And the aging process continues and AI Jessica changes again. And, it keeps on going. She changes again and she changes again. And so this is fundamentally one of the most important processes that we can understand molecularly about aging. And with that, this is where Steve Horvath came up with this idea. Can we measure someone's DNA methylation and actually estimate their age? And the initial clocks of these were done just on chronological age. But after he was able to show that, yes, you can actually do that remarkably well. You can correlate DNA methylation to chronological age. We then started becoming more sophisticated, and we started developing other types of aging clocks. Some of them are really positive, like grim age, right? Estimating your time till death that you can invert to get your biological age. But there are other ones here that are really important in different ways. And this is one of the ones I actually like the most. It's called Denis and Pace. And it's the rate at which you're aging, right? So so think of it as a slope. If you're aging at the expected rate, your slope is 1. And one of the reasons I like this is, let's say I've been really bad with my health my entire life. And so I have a really high biological age, so let's say my biological age is 60, but then I decide to start having good habits. Now, if you notice, when you start having good habits and eating well and exercising more and this and that, do you notice how you start feeling better? And that feeling better actually happens relatively quickly. Well, your biological age is probably going to stay at 60 for a while, and getting it to go down at all is going to take a long time. But what's actually really cool is you will see the rate at which you're aging change remarkably fast. Like within probably a month, you'll see the rate change. And so this can be really encouraging, right? So you can have someone who has a higher age, but their rate of aging is slowing down, and even reversing. So that is a really positive thing. So that's one exciting thing. But one of the things my group was interested in looking at is, okay, now we look at this whole concept, and what I've shown so far doesn't incorporate the important point we talked about before, which is health span. We can look at lifespan, but can we look at health span? And so we developed an aging biomarker that looked at not only time till death, but really incorporated the quality of your life into how we were measuring biological age. Not only that, we decided to do something else different. Well, DNA methylation is super important for aging. There's a lot of other molecular data that tells you a lot of other really important things. So on top of DNA methylation, we added in other molecular data. So we added in proteins and small molecules to create our measure of biological age, which is called omic age. And here you see we added clinical labs, DNA methylation, proteomics, and metabolomics. And the really nice thing about this is it incorporates a lot of information. The really bad thing about this is if you are practically to measure something like this, it's sort of a disaster, right? No one is going to want to get 10 blood tests. Like each one of these is going to cost a lot of money, so it's completely impractical. So to address that, one of the things we did was we generated the clock using all this different type of molecular data. We translated all of those different types of molecular data back into DNA methylation. We recoded everything in DNA methylation. So now we have a framework that we actually have estimates of important metabolites, important proteins, but we've created linear models of each one of those, a DNA methylation, so that we can create the entire biological aging clock with just DNA methylation. And the really important thing about doing this is when you're incorporating other types of molecular data, now I can have a picture like this, and I can measure your biological age, but I'm not just telling you what your biological age is. I can give you a lot more information about your physiology. I can give you information about different inflammatory pathways, endocrine function, immune function, et cetera, all with just DNA methylation because it's been informed by other clocks. So this concept of looking at multiple types of molecular data together is a really important way to understand aging. And when we do this and we actually just look at things like mortality predictions, right? What's actually pretty impressive is we have 10-year mortality predictions. These are our two clocks that are close to 90%, which is pretty good in terms of actually capturing things like longevity. I would be remiss if I didn't also say that this was an episode of the Kardashians. If you want to watch it, there was some drama because Kim was upset that Khloe's biological age was younger than hers relative to her actual age. So a little fun fact there. So just in looking at this, when we look at aging biomarkers, we can measure these with epigenetics and other metrics, and we can get a lot of information about our overall biology right here. You know, my age, this is actually my biological age, and this is actually my rate of aging. And so I'm aging really well. This is probably higher than I feel like it should be because I probably don't sleep enough and I probably don't meditate enough is really where I think I'm having trouble. So that's DNA methylation in aging clocks. And so I just want to give an idea of how each one of these contribute. And the other one I want to highlight here, because I think it's incredibly valuable and it's amazing what we can do, is sort of looking at what is called the metabolome. And what is the metabolome? The metabolome and metabolomics, that is known, is measuring the small molecules. And I want to talk about why I think the metabolome is particularly important in terms of inferring information about aging. And so the metabolome, what is it? It's measuring all the small molecules that you have in your blood. So this is actually really cool, and it's actually remarkable to think that you can do this. This is not a Theranos thing, I promise, but you can take a small amount of blood and you can measure lipid levels, blood sugar, amino acids, hormones. But not only that, you can capture all sorts of things about functioning of different energy pathways, different aspects of function from serotonin receptors, et cetera. But beyond that, you can actually capture a lot of other really important things that you may have no idea even you may have in your system. One of them is environmental toxins, and we can definitely get good profiles of medications, nutrition, et cetera. And so you can think of this, okay, you have this, you can capture cortisol, blood sugars, and then really these sort of comprehensive metrics of a lot of other relevant exposures for aging. And that's why I want to bring this up. So I'm just going to give you an example here, because I actually don't think I would have believed this if I haven't seen it as robustly as I have. How well can we actually measure dietary patterns from blood? And so what we did is we actually took blood on a lot of these people in our biobank, and what you can do is you can look at this in combination with a lot of food frequency questionnaires where people have reported things, and you can look at it with different chemical annotations and food data libraries. And what we've actually found is the best way to actually capture these nutritional profiles is not a specific food. Like, I cannot go around and say, oh, you had a brownie or you just had a cookie per se. But if you look at the chemical components that are in that, that is easy to capture. So it is much easier to capture categories of foods than actually a very, very specific food. But I would actually argue that is most important, because if you think, like, on a daily life for me, I have fruit every day. It may not always be the same fruit, but I pretty much probably have around the same amount of fruit every day. And so that profile stays relatively constant if you're looking at key metabolites. So we can take blood and we can actually look at all of the molecules and annotate them to these different foods. And when you do that, you can actually look at these categories and you can look at them across something like, this is our measure of biological age here, omic age, and you can look and see what's most protective against aging and what's most associated with positive aging, like highly processed foods, juices, sugars, sugar-sweetened beverages. And you can do that and show what's statistically the most robust, et cetera. But beyond that, what is even more amazing, and I think is probably even more critical, is can we actually look at the scope of this and look at it for biological aging and generalize that to across diseases. And for people who have ever seen like RNA sequencing data or looking at things like UMAPs, this is what we can do that I actually think is super cool. This is a UMAP that is capturing the interrelationship of all these metabolites across 80 different diseases and biological aging metrics. And it's sort of capturing them and relatively rank ordering them. The darker green means, in this case, it's associated with decreased aging. And the higher red is associated with increased aging. So, and then we have annotated on here the different food categories. So what we can see here is, right, this huge tail here, all sugars, like processed sugars. And you can annotate things here like animal diets, refined grains. You see down here fruits, vegetables, healthy oils, and then nuts, seeds. And then I actually want to point out, I always say this now, ergothioneine is like my favorite thing. Does anyone know what ergothioneine was who wasn't at the lecture this morning? Mushrooms. Eat your mushrooms. I have seen this result over and over again. So ergothioneine is really quite amazing. But like, you can actually then also do it where you keep the same construct and then you look, and you can just highlight the associations for each disease separately to sort of say, okay, where does this metabolite live relative to all diseases, and how does it affect cardiovascular disease or COPD or depression? And so it's a nice way we can generalize overall effects of different metabolites on disease and aging. Okay, so I want to highlight that because I think it's really critical to get at a lot of areas that are probably underappreciated with the metabolome, particularly things like nutrition, where we can look and we can actually sort of quantify profiles. One of the best ways to actually do this is to look at eating patterns. So like looking at how you eat according to like a Mediterranean diet, or you can do a bad diet. There's like 8 or 5 different dietary patterns that have been really well characterized, and then give it suggestions of what needs to be improved. But then I want to just highlight these two because some of these are things that are often hard to capture. And we can actually really look quite comprehensively at a lot of exposures. And some of these are actually pretty important because sometimes there are cumulative exposures that we may not even know that we have. Now, it's super easy to see things like smoking tobacco, smoking weed. I've done studies in them. You can tell it's super easy. But the other ones I think that are really coming up are some of the environmental toxins and also air pollutants, like that we can actually capture quite well and get a sense of how these are impacting aging. And a good example here is like PFAS, right? PFAS is the plastics that are in things and their impact on aging. Okay, so now I've talked about 2 areas where I think a lot of what's happening in aging, there's a lot of stuff going on, but there's a lot that has been done. And the area I want to focus on for the rest of this is the area of antibodies. And part of the reason I'm so interested in this is because I think it's a very understudied area. And so just looking at antibodies and antibody reactivities and how that affects aging, I think there are many underexplored questions here, and I love chatting about this even more because I think there's so many important things we can think about. But the bigger premise of why antibodies, like why is that even important in aging, you could ask. And it's really this immunologic process, right, where we have a viral exposure or whatever, we have immunosenescence where there's dying of cells. But as we age, right, we are not able to clear those cells as well, so we get antigen accumulation. And through this process, we often get loss of self-tolerance and the development of autoantibodies to ourself. And this process is a process where, as that happens, that causes a lot of, that affects a lot of the different hallmarks of aging in this physiologic process. So this concept of the rise of autoantibodies and how that may impact aging is something I had a lot of questions about. And I think there's so much to learn here that I'm saying this is like a missing omic, right? So I talked about DNA methylation and metabolomics. Those are called omics, proteomics. But people really haven't talked about the antibodies in this framework. And I think it's perhaps one of the most important missing links that we have more to understand about with aging and disease. So in talking about this, I just want to talk about this. I'm going to pull back to biology. We have a lot of people here doing amazing research in this area that I'm super excited about hearing about today. But really even just from the beginning of, okay, if you think of even COVID, right? If you think of what happened during the pandemic, people got COVID, and then there was this post-acute long COVID response, right? And what's happening there, and why are people developing these symptoms of fatigue, et cetera? And one of the ideas is that when we have infections, right, often our body, again, and going to try to react to develop antibodies against them, develops these autoantibodies. But can we actually characterize that? And how well is that characterized? And this to me was a fundamentally important question. Can we even just characterize in any way how different viral exposures are associated with autoantibody reactivities? And because this would be a first step in aging. So one of the things we did was we looked at a bunch of people in our biobank, and we looked at exposures to several different herpes viruses. And then we just wanted to ask the question, can we identify autoantibodies that they are associated with, right? Because this would give us an idea of the background biology. Now, this plot may look hard to read, but I'm going to explain it so you can see what this is. This is called a Manhattan plot. And there's different herpes viruses, herpes virus 1, simplex 1, 2, we keep on going along, CMV, I'm highlighting some of the most important ones, Epstein-Barr, which is mono. So these are the different herpes simplex viruses. This is a Manhattan plot, and this is a negative log of the p-value. So a p-value up here of 80 is a number of 80 is a p-value of 10 to the negative 80. And these are autoantibodies associated with that specific herpes virus. So what we're seeing here is we're looking at this in about 1,200 people, and we see very clear signals of certain autoantibodies that are really strongly associated with different herpes viruses. Now, I just want to point out here, that dotted line is a line of statistical significance that's like 10 to the negative 6. Look at the number of things we have all the way up to 10 to the negative 20. There's a huge amount of associations here of autoantibodies and herpes simplex viruses that we aren't even capturing here. So when we look at things like this, though, one thing I'm always thinking is like, I typically don't believe things unless I see them really robustly. And the best way to do that is always to try to do the experiment again. So we did it again in a much smaller number of people, but what's so striking is that we saw the exact same autoantibody viral associations. And to me, this was absolutely a level of like, wow, what we're seeing is likely real, that these autoantibodies are associated with these viruses. Okay, so that may be like step number one in saying, is there any type of potential causal relationship between these viruses and the autoantibodies? There's a lot more steps to go through, and while there's several that I can't do, there are some that I can. The next thing that we often do is, okay, if we see this really strongly associated, can we actually take the viral peptide and can we actually predict that autoantibody, like whether or not someone has it? And so we did this. And we looked here and we said, okay, like let's take the PHLDA1 autoantibody, and can the herpes peptides that were associated also predict that someone would have this? And if you see, the top line in each of these cases is the viral peptide that was most associated with that autoantibody. And when we do this in a training situation, we see good prediction. But what was amazing to me is usually when you do it again and you try to test something, usually in the testing set, your test reliability goes down. You don't have as good accuracy. In this case, our test accuracy went up in every case, suggesting that we really can predict these, and you really can take the viral peptides and predict almost perfectly whether or not someone's going to get the autoantibody. And that to me was really exciting. But beyond that, right, the next step that we can take is, okay, if a virus, is somehow inducing an autoantibody, very often they'll have similar sequences similar viral sequences here when we're looking at the peptides. And if they're the same, that would suggest that, it's likely that your body thought it was one thing because it sort of folded the same way, and then it tried to make it, but it made it to the wrong thing. That sequence homology is really important. And what we could see here is that in many cases we had very good sequence homology, really suggesting that this concept that viruses could induce different autoantibodies and we could really identify them specifically, I think we can do on a much bigger level than what we are. And well, these are only the first steps. It's certainly exciting to see that we could start understanding this more. Okay, so that's great. So, okay, we can sort of see, I think more and more work, we can understand how different autoantibodies arise. We might really be able to look at that in a lot of different ways. But what about aging? Let's get back to aging. So the first thing we asked in my group is like, okay, can we create an autoantibody clock or an autoreactive clock? How well are they predicting biological age or predicting death? Well, this is step number one. This is pretty darn impressive to me. These are associations between age and predicted mortality and the accuracy. This is a training set. This is a testing set. These are remarkably accurate. So these are as accurate, I was looking this up, is almost some of the best clocks out there. We can predict time till death here with an accuracy within it's within, I think, like the the MAS, if we take that's the square of that, the square root of that, that is the number of years, are quite good here. So we're able to do this remarkably well with not that many autoantibodies. So they're great predictors of time till death. But can we really understand other things about these autoreactivities and how they associate with biological aging? And this is where we wanted to look at this in multiple cases. So now we went over some of the biological aging clocks. If you remember, we talked about Dunedin pace, which is the rate, Grimm age, which is time till death, omic age, which is the one we made as multi-omics. We also have on here one that is a clock that was called retro clock, which is really based on a lot of underlying biology of like genomic stability, cellular senescence. And pheno age is sort of a little bit like omic age, but not as good. So these are 5 different clocks. Okay, they can all give you they all are correlated, but they're all giving you different bits of information. Can we look at these autoantibodies and see how they relate to these measures of biological age? This is just a volcano plot of all the p-values. There's not a ton to show here, other than, again, the way you would read this is a p-value, and these are the beta coefficients. These are really, really close because those are, again, the slopes, which are really close to 1. But the primary thing I initially was asking is, if we look across these aging clocks, if I'm going to believe that there's an autoantibody that actually is related to something like aging, it should be pretty consistent across the clocks. And do we see that consistency? And the answer there was yes. A large majority of the autoantibodies had concordant associations across the clocks. But this is where things got really, really interesting. Okay, so I want to give you an idea of how to read this. Each of these dots are the different measures of biological age that someone has, okay? This circle is a circle of autoantibodies that we see related to aging of these clocks. Lines or edges that go from one of the autoantibodies to the clock are blue if it is protective against aging, and they are red if they are associated with increased aging. The outer circle are diseases, and we're going to talk about that a little bit more later. But the first thing we see here is like, number one, we can see deleterious autoantibodies associated with accelerating aging. But this was a big shocker. Look at the number of protective associations with biological aging. Like, this to me was really mind-blowing because I had no idea, I didn't think anything like this would exist, that there'd be this number of potential protective associations of autoantibodies against aging. And in many cases, you see these associations with diseases too, protective associations. So that was really interesting. But the other thing that was actually really remarkable is actually if we looked at these autoantibodies and we looked across the hallmarks of aging, there are like these physiologic processes of hallmarks of aging. What we could actually do is we could annotate a lot of these autoantibodies to different hallmarks of aging and specific mechanisms that it looks like they could be impacting. And about half of them were associated with increased aging, a half with decreased. And these were ones that were common among clocks. So we could actually see that we have like almost a yin-yang, that there's certain things we see increasing aging, certain things we see decreased aging. And then there are certain cases where we saw complementary mechanisms going back and forth. And one of the examples I'll give of this that was really interesting was the contrast of sort of mechanism of complement, innate activation, and pro-inflammatory aging, that we saw antibodies really increasing aging, and in contrast, sort of this humoral homeostasis, anti-IgE antibodies that were associated with decreased aging and sort of just overall homeostasis of the humoral system. And this is really interesting because I'm not actually sure how much these type of things have been characterized, particularly the protective nature of some of these autoantibodies in terms of sort of just cleaning up and maintaining homeostasis. So, but beyond that, one thing that was also interesting that I'll point out is while there's a lot of mechanisms that were the same, when I was looking at autoantibodies associated like distinctly with certain clocks, one thing that was fascinating is a lot of the mechanisms that were associated with a specific clock were also associated with how that clock was developed. Like, for example, rate of aging was associated with a lot of things of like rate of repair and recycling. And it looks like I literally copied how the clock was developed with what the autoantibodies were, but I swear I didn't. I was so impressed that so many of the things for like the retro clock really were autoantibodies it was detecting based on things like cellular aging, DNA repair, and cellular senescence. So this is really exciting. So just getting at this whole point, though, that we can look at these clocks and we can look at it with these underlying biology and understand a lot of new things. But I want to get to a few other points that sort of were really interesting questions. One is, do we see differences by sex? And I'm trying to think of even how I got on this because it wasn't really something at the forefront of my mind. But the bottom line is, yes, the autoantibody rectum is not neutral gender, not gender neutral at all. And that we see several common mechanisms. There are certain things that really seem to be specific. So blue is things that are common, and these are things that are not. And now, I showed you these plots before. Now, I'm just dividing it into males and females. And what you see here is that, right, a lot of what we saw before sort of looks similar to more the female plot where, okay, the females have overall more associations, which is something we do know that females tend to have more autoreactivities. And while then the adverse associations or things associated with accelerated aging seem to be about the same, the protective component was really, really driven by women. I mean, and when we look at a lot of things like ovarian disease, myocardial infarction, chronic kidney disease, cancers, these components are not, they're important for disease, right? And so this to me was striking. Now, when I recently gave this talk at another conference, one of the questions people asked was, okay, is that just because of menopause? And so, you know, is it hormonally driven? And so we're looking into a lot of these things now. One of the things is, no, it's not hormonally driven, that we see these protective associations before and after menopause in women. There's a lot of other questions we're looking to answer here, but I think this is certainly begging the question about important things that these autoantibodies may tell us. But on a bigger note, one thing we can do is we can actually classify mechanisms that seem to be stronger in men and women and what are common. And in women, there seems to be much stronger sort of epigenetic regulation, and humoral homeostasis seem to be much more female driven, and then proteostasis seems to be a stronger component to men. So really suggesting that there's more I think we can understand about aging in a sex-specific way. Okay, so now we talk about autoantibodies and aging. And I think there's a lot to uncover here, and I think we're just beginning to unpack the surface. But the bottom line is I think there are a lot of very important things here that we can understand about aging. But in looking at this category, I would be remiss if I didn't talk about some of this in the context of age-related chronic diseases, because in many ways, I would say that's where I'm seeing like the most potential impact. So when we look at these autoantibody reactivities and we look across electronic medical record data, again, with aging, we see, this is what we see with aging, but if we go here to this outer circle, one of the things I'll say is a lot of the aging signatures are very, very much the same direction as we see for specific diseases. But if we look at these across specific age-related diseases, and here I have cancers, I have some immune diseases like inflammatory bowel, and then I have neurodegenerative diseases here. I want to give you a concept of what you're looking at here, because I think you need to understand that to appreciate how amazing, how crazy this is when I saw this. In each of these cases, these are people who have not yet gotten the disease. So when we have here people who are have colorectal cancer, the cases we're looking at are people, this is before symptoms, right? Before Parkinson's diagnosis or Alzheimer's diagnosis. And we see single autoantibodies with associations that are not only higher than like for Alzheimer's, like the gold standard is, do you have ApoE4, right? If you have ApoE4, that's like your biggest risk factor for Alzheimer's. That's way down here. If you think of like the causative relationship between smoking and lung disease, right? That's like the gold standard in epidemiology in terms of showing something as causal. That association is 20 to maybe 30 max. Like, look at how strong these associations are for single autoantibodies and diseases. And this to me just really blew my mind. And I'll give you a few examples digging into this just a little bit deeper. Alzheimer's disease is an area that's had a lot of like expansive discovery in the last 5 years. And a lot of it is around certain proteins like the accumulation of p-tau protein. And there's a lot of work even going into treatments. Well, one thing I'll say is when we look at a lot of the proteomic data and we're looking at these proteins, often we're only capturing a subset of the total proteins that exist. One of the great advantages of these autoantibodies, now autoantibodies are targeting proteins, so each autoantibody is associated with a protein, is we look at about 3 times the number of proteins targets with autoantibodies. So when it comes to something like p-tau, which is, where there's a lot of research going on with Alzheimer's disease, these are all the different types of categories and where our autoantibodies could be affecting p-tau. And we see these associated with Alzheimer's disease in our studies. Not only that, when we look at something like p-tau, right now, I think there are several clinical trials in preventative treatment for Alzheimer's disease, is giving people the antibody to types of p-tau. This is one of the treatments ongoing. In our study, we see natural tau antibodies are associated with reduced risk and protection of Alzheimer's disease, particularly in females. So in essence, we're sort of showing in a body, right, that the same sort of things that they're trying to look at in terms of treatments is actually working in nature, potentially in some people. Really sort of highlighting the potential translation of some of these things into potential treatments. And a little bit further, just looking at this with Parkinson's disease, right? Parkinson's disease is really probably at this point more complex than Alzheimer's because we don't have as many biomarkers for it. The treatments are not as clear. And I want to show you this because right here are sort of risk factors for Parkinson's disease. I said this in the last class, I'll say it now. When we look at risk factors for Parkinson's disease, does anyone know why the golf course is, it says on this. The golf course is up there. It is pesticide exposure. And so that's a key risk factor for Parkinson's disease. These are the set of top autoantibodies we see associated with Parkinson's disease. again, just the magnitude of this in people who are asymptomatic. So again, think about the idea of health span. We want to be intervening early, right? If we can intervene before we are seeing the physical symptoms of disease, that is super important. And when we look at things like this, I always want to look at things like, are related diseases showing consistent patterns? So for things like Alzheimer's and Parkinson's, are we seeing it in vascular dementia? Are we seeing it in MS? Are we seeing it in ALS, et cetera? So what I have here, the top hits for Parkinson's disease, and those are in this orange color, and then these are related diseases, and we still see like these protective effects. These are all nominally significant results across. Each one of these is an autoantibody. I don't have them listed right now for proprietary reasons, but we see consistent associations across these diseases. But beyond that, when I looked at these hits, there's so many things that have relevant mechanisms that would suggest there might be ways we might be able to think about treatment. And I am not the expert in the room when it comes to engineering antibodies, but this is something I'm really excited about, because when you have a hit like this, which this is a gene that causes Parkinson's disease. If you have inactivation of this gene, you will get early onset Parkinson's disease. This is one of our hits with autoantibodies. It does beg the question, are there simple ways we might be able to intervene, even through things like immune modulators, or other ways we could develop agonizers or inhibitors of this. But it just definitely puts you right toward potential prevention or even delay in outcome. And so those are some examples I'm seeing with neurodegenerative. And I'll just say, we have yet to look at other areas, but cancer is another one where we're, again, seeing these really similar patterns here that are really interesting. And in this case, blue means these are people who have not yet gotten the disease, but get it. Red are people who have the disease. So we often see the same autoantibodies in both, which we'd also expect. And to me, when I'm looking at this, there's just a lot of potential areas for low-hanging fruit. And this is a new area to really explore for aging, and definitely for translation, because looking at monoclonal antibodies, a lot of those therapies are really one of the primary areas of focus for a lot of new drug development. So when I think of this, and I think about antibodies, I'll say, I can say, okay, we can look at different disease risks, but I think really when it comes to aging, there's so much more that we can think of. We can also, again, think of, I think, different types of ways we could develop monoclonal antibody treatments that I think will really help. I think we're just really at the beginning of understanding these things. And for the people in here who are virologists, the one thing I didn't show in this talk that I show in a lot of my other results is that when we look often at the viral exposures, we see the viral exposures very often that, like HSV, that are associated with a lot of these neurodegenerative diseases. Can we actually look at those viruses, understand how that may be instigating autoantibodies, and how is the disease developing? Is it both of them? Is it one of them? And I think that's an area that I'm really excited to be looking into more. Okay, so we're at the end of the hour, but I would be remiss if I didn't say something about AI, because I think it's a topic now of almost every conversation that happens when you're doing anything, right? It seems to come up a lot. So let's just think about AI and AI in the space of this field. And as you know, there's a ton of potential, I think, for AI, and there's a lot that we can do in this field. And I think the one thing is, with all of this data, there are a lot of ways to train large language models to really inform us in ways that are beyond what we can do now. And my lab is actually working on developing a foundation model right now. And it is so interesting to work with people who are real experts in how to do this, because there's so many ways and things you think about, but putting a lot of this type of data in, training algorithms to sort of try to help understand. And I think this is sort of, a vision of how would this enable future treatment of disease. I think there's a lot that we can do here. And, even envisioning things where likely the best preventative framework may be one where we are, every 6 months sending in a sample, getting things measured for risks, and have that as a preventative framework going forward, right? That type of framework may end up helping us intervene early. But as much as I think there's potential there, I also want to highlight the other point, which is the humanity of what we're doing, the human empathy of treating disease and working together to optimize health. And that I think that we can't, the idea of AI is something that can help, but it's never going to replace the human. And so with that, I'll just end my talk and say, I think there's a lot of exciting things happening here, a lot of different types of multi-omic molecular data that are informing us on health, showing us different ways we can go. I think thinking about health span, and if you're ever thinking, should I go to the doctor or should I work on this now? The answer is probably always yes, because that's the preventative mindset. But also really highlighting this area of antibodies, viral antibodies, autoantibodies, and how those may help inform aging, inform disease, and potentially inform treatment, I think is a really exciting area. And so with that, I'll thank several people in my group here for all the work that they've done on this, particularly Shirley, who did a lot of the aging stuff, and Nicole and Sang-Hun, who did a lot of the viral autoantibody stuff. And with that,